It is my pleasure to introduce Gergo Szanda, and Dr. Szanda is a visiting scientist at IU Bloomington, and he has kind of a special interest in something that we were interested, at least from our nonprofit as well. So in years past, we've done some work with the patient groups looking at the use of CBD and how patients have been using it beyond the Farm Act bill that was passed and made it explode here in Indiana. At least in our initial studies, we saw a lot of patients, up to 50% of patients had used CBD as a way to mediate or mitigate symptoms associated with this disease. So we started asking more questions. The problem is the literature is pretty weak in terms of their use in any kind of randomized controlled trials, obviously. So we turned to the science, and this is where I asked Dr. Szanda maybe to talk about this. He has dedicated expertise. Now, you do work in adrenal steroid production as well, but also the role of endocannabinoids, and he'll tell you a little bit more about what those and how they work in terms of metabolism and other types of tissue injury. So please join me in welcoming Dr. Szanda. Thank you.
Thank you very much indeed. Good morning, everybody. I wish to thank the organizers for inviting me. Although my current work focuses solely on basic research, I was also trained originally as a clinician, and it is always a privilege to talk to patients, to be able to talk to patients. And today I wish to speak to you about cannabinoids in general, endocannabinoids, or the body's own cannabinoids, and phytocannabinoids, cannabinoids that are found in the cannabis plant. And before we do that, this is actually my overview, and then after introducing cannabinoids, I would like to talk about the very recent knowledge about their involvement in liver disease, their possible involvement in liver disease. But first I have to make some statements or introductory, what's the proper word for it? It's the disclaimers nowadays. What I'm going to talk about is preclinical data. So none of this should be, extrapolated directly into the clinical practice, and certainly shouldn't be regarded as treatment recommendations. And for the sake of a broad overview, some oversimplifications were necessary. So what are cannabinoids? First of all, what are endocannabinoids? So even though mankind had been using cannabis and cannabis products for thousands of years, we know that for sure, it's fairly recent knowledge that our own body is able to produce cannabinoids, and these are called endocannabinoids. As a matter of fact, most of our organs, the cells in our organs, have the capacity to produce endocannabinoids from membrane lipids. Now, these endocannabinoids are produced under certain circumstances by dedicated and well-controlled enzymes, but nonetheless, the cells can produce the two main endocannabinoids. One of them is called anandamide, and the other is 2-AG, or 2-arachidinyl glycerol, but it's usually, you'll see it in the literature as 2-AG. The first one that was discovered 25 years ago is anandamide, and the name comes from the Sanskrit word for joy, and 2-AG is just a real plain chemical name. Now, what do these endocannabinoids do? They are lipophilic molecules, so once they are produced in a cell, they will be released into the environment, and they will modify the function of adjacent cells. You can think of them as local hormones. They don't really get into your bloodstream in high concentrations, but are able to modify the function of cells that are close to those cells that produce these endocannabinoids. And the effect of endocannabinoids on these adjacent cells will be dependent on what sort of receptor that pertinent cell has. And strictly speaking, we have two endocannabinoid receptors named CB1 and CB2 receptors. These are abbreviations for cannabinoid type 1 and type 2 receptors. Their function is to recognize the presence of endocannabinoids and then modify the function of the cells which possesses these receptors. The physiological function of CB1 receptors is way more studied and much more elucidated than those of CB2 receptors. We know that CB1 receptors are mainly expressed in our brain, in our central nervous system, and when they are activated, they can modify many complex brain functions. They have a pleiotropic or multifaceted effect in the brain. What are those? We know that cannabinoids via the CB1 receptor can affect our appetite. They affect our energy metabolism, our total energy expenditure, but they can also modify really high CNS or central nervous system functions like our mood. They can modify our complex motor function, the way we perceive pain. But CB1 receptors are also present in some peripheral tissues, and they can modify how our body processes fats and how it stores fats, and I will get back to it, to this aspect shortly. Now, the function of CB2 receptors is way less clear. It is a bit more elusive. We know that CB2 receptors are expressed mainly in immune cells, T and B cells, and macrophages, that is. But what CB2 receptors do under physiological conditions is somewhat unclear. We think that their main function is immunomodulatory, so we think they somehow modify complex immune functions. And we know from human populational data that certain mutations of CB2 receptors are associated with higher risks to develop some diseases. For instance, higher risk to develop cirrhosis in hepatitis B, or in children who carry certain mutations of CB2 receptors, we see a higher incidence of non-alcoholic fatty liver disease. So from that, we can infer that it possibly modifies immune functions, but we don't know how and why, so more research is needed on that. So these are the endocannabinoids. You can think of them again as local hormones that usually modify the function of other hormones or neurotransmitters or cytokines or immune modulators. There are also plant-derived or so-called phytocannabinoids, and these are the cannabinoids that we had been using way longer than actually endocannabinoids. What are phytocannabinoids? Phytocannabinoids are biologically active compounds that are found in the cannabis plant. Now, the cannabis plant is a gold mine, a pharmacological gold mine, and also a pharmacological mess. Because the cannabis plant, it's really, there is no method in the madness that's in the cannabis plant. It has more than 100 biologically active compounds. I only named a few. Both the names and the abbreviations are really confusing. And we have only just begun to understand how these molecules can affect physiological or pathophysiological functions. But I wish to really emphasize just two of these compounds because these are the two main constituents of the cannabis plants, one being THC or tetrahydrocannabinol, and the other is CBD or cannabidiol. You may encounter these molecules a lot on the internet, and I will come back to that. But the take-home message is these are the two main constituents, and THC and CBD convey most of the classical effects of cannabinoids on our body. So what do they do? What are the classical effects of these major phytocannabinoids? So let's start with THC, because we know a lot more about THC than CBD. THC, when it gets into our system, it will use the same receptors that endocannabinoids deploy. So THC will bind to CB1 receptors. If a cell has CB1 receptors, it will be sensitive to THC. And since CB1 receptors are mainly found in our brain, THC will exert its effect mainly in the brain. It is a psychoactive cannabinoid. And when CB1 receptors are activated by CB1 receptors are activated by THC, we can see increased appetite. THC has some analgesic effects. It can be anticonvulsive, so it can suppress, to some extent, epilepsy. It has weak anxiolytic effects, and it also can be antiemetic, so it can work against nausea. Now, none of these effects are particularly robust. However, the combination of such favorable effects justifies the medical use of THC-containing compounds in some diseases, for instance, in some cancer-associated pain, or to mitigate the side effects of anti-cancer therapies. However, these favorable effects come with a price, namely THC via CB1 receptors is probably pro-inflammatory. It can cause an unwanted hypertension. It can change mood and cause mood alterations in the long run, reduce motivation, etc., etc. These side effects are well documented in the cannabis abuse disorder, on which I can elaborate for the sake of time. But the gist of my saying is that besides the favorable effect, you will have some side effects. Now, how THC works via CB2 receptors is much less known, and it's quite debated. It's probably anti-inflammatory, but I put a question mark here because the data is very confusing and very weak. Now, the other main constituents of cannabis and cannabis products is typically CBD. And CBD, funnily enough, exerts its main effect not via the cannabinoid receptors. It will act via other receptors. The names aren't really important for us right now, but we know that CBD has a very strong effect against epilepsy. As a matter of fact, Epidiolex has been FDA approved to treat some specific forms of childhood epilepsy. So this is FDA approved, this is good data, this is strong data. So we know that CBD is anticonvulsive. The other claimed effects of CBD, like analgesic or anti-inflammatory data or anti-inflammatory effects, are not as well established as the anticonvulsive. And importantly, CBD will actually modify the way THC acts in our body. And it is believed, although it's not supported by very strong data, that CBD will suppress the psychological effects of THC. So, as a rule of thumb, you could say that the higher the CBD content of a cannabis product, the less neuropsychological effects you should expect. Okay, now after this really technical and long introduction, I'd like to share some data on cannabinoids' effects on liver disease. And again, I will focus not on the treatment of symptoms, because as we have seen, some effects of THC are really well suited to suppress symptoms of many diseases, anxiolytic or analgesic effects. Those can be exploited to treat symptoms. But here, when discussing liver disease, I like to function on curative effects. So the question is whether cannabinoids have an effect on the progression or development of the disease and not on the symptoms. And non-alcoholic fatty liver disease and alcoholic liver disease is where we have the strongest evidence showing that blocking the endocannabinoid system has actually beneficial effects. How do we know this? We know this because we can model these diseases in animals. If you feed mice high-fat diet, you can actually make their liver accumulate a lot of fat that is reminiscent of the human non-alcoholic fatty liver disease. And also, if you feed mice ethanol, which they are usually not willing to consume, so you have to use some sort of trickery, but you can make them consume ethanol. At the end of the day, you can see here ethanol, similarly to high-fat diet, will cause lipid accumulation in the liver. Now, that is usually problematic because lipids themselves can be directly toxic to liver cells, and also liver accumulation makes the liver more susceptible to other noxious stimuli. If, under these conditions, you give a drug called rimonabant that blocks CB1 receptors and blocks the effects of endocannabinoids, you can see that you can lower the fat accumulation in the liver, and that is also accompanied by a drop in the transaminases. So you can actually protect the liver from the harm that was posed by high fat or ethanol. Now, please ignore all these molecules. They are all very technical. I just wanted to demonstrate that we know exactly how the blockade of CB1 receptor exerts this effect. We know which molecules are the sites of action. So this is really strong preclinical data. And you might ask, why don't we then use rimonabant in the clinical practice? We did. I don't know whether you know about this, but rimonabant as a CB1 blocker, an anti-cannabinoid drug, was marketed in Europe for the treatment of obesity, metabolic syndrome, and type 2 diabetes. And it was really effective in treating those conditions. Patients loved it, clinicians loved it, until in phase 2 trials it turned out that it also penetrates the brain, and it turns out blocking CB1 receptors not in the liver, which was favorable, but in the brain has serious side effects. Namely, it increases the risk of suicide and suicidal ideation, and when this was documented, it was withdrawn from the market in 2009. It was a huge blow to the entire field of cannabinoid research, from which actually this endeavor has not bounced back, I'm sad to report. But this really exemplifies that a pharmacological effect in one organ may be favorable, and the same effect in another organ may be actually risky. What about autoimmune hepatitis? Now, there are some preclinical studies on the effects of cannabinoids on the development and progression of autoimmune hepatitis, but only a few. You can actually model autoimmune hepatitis fairly well in mice by injecting the drug called CON-A. And I don't know how familiar you are with histological sites. This is a control liver. It all looks nice and healthy. And when you inject this drug to mice, you can see these areas that are infiltrated by a lot of immune cells, and you can see necrosis and apoptosis, so cell death going on. And this is T-cell mediated, just as autoimmune hepatitis is in humans. And if you apply CBD or THC to these animals at the very beginning of the pathological process, you can actually suppress the T-cell involvement and liver damage. And that is also corroborated by the drop in transaminases in these animals. So whereas in control or injected animals, you see these large affected areas, you barely see those if THC or CBD is present. Now, there are literally no follow-up to these studies. These studies didn't progress into clinical phase, and I must admit I don't know the exact explanation for that. But one of the reasons may be that while at the beginning of this disease, autoimmune hepatitis, the activation of cannabinoid receptors with CBD and THC may be beneficial, that may not apply later on when cirrhosis develops. And this is actually a study we published when I was working at the NIH. If you induce fibrosis in animals, you can do it either with inducing cholestasis or with CCL4, and in red you can see the scar tissue in the liver. Control animals don't have any scarring, and affected animals do. At this stage, not activating but blocking the endocannabinoid system, again with rimonabant, is beneficial. And we know again how the blockade of CB1 receptor actually reverses fibrosis. So that demonstrates that blocking or activating the cannabinoid system may have different effects at different stages of a disease. So you have to be very careful when extrapolating from preclinical data, and that is probably why the preliminary and optimistic data in autoimmune hepatitis had no follow-ups, because at the stage of fibrosis, cannabinoids may have the complete opposite effects as at the beginning of the disease. And this is my most important slide by a long shot, because this was all very technical. But I'd like to point out that if you hear any claims about cannabinoids having beneficial effects on any human conditions, you should be very skeptical about the data, and unless it is really backed up by solid evidence. If you hear anecdotes about cannabinoids' harmful effect, you should take it seriously unless proven otherwise. And cannabinoids are natural compounds, and there is this halo and mystique around anything that is deemed natural. Most of nature's most potent toxins that will kill us are natural compounds. So just because it's natural, it doesn't mean it's not dangerous. So please keep that in mind. Different strains of the cannabis plant will have different cannabinoid content. The marijuana that one might acquire on the streets or legally has a lot more THC these days than it had 30 years ago. And that poses a huge problem, and there's a huge literature to this, but you have to keep that in mind, that the marijuana of these days is not the marijuana of the 60s. If you have liver disease or concurrent medication, that will affect the metabolism and then eventually the effects of cannabinoids that you take, and the other way around. So that needs to be also reckoned with. And as we saw with autoimmune hepatitis and non-alcoholic fatty liver disease, each condition is unique and no universal statements about cannabinoids should be made. We saw that blocking cannabinoids in non-alcoholic fatty liver disease had a beneficial effect, whereas activating the cannabinoid system had a beneficial effect, at least at the beginning of the development of autoimmune hepatitis. And finally, cannabinoids may affect subjective symptoms and the real progression of disease differently. So cannabinoids may be still usable to treat and address symptoms, but may not be usable to affect the progression of the disease itself. So as a symptomatic therapy, they may be warranted, whereas as a causative therapy, they won't necessarily work. And finally, what we need is a lot more controlled, solid preclinical and clinical evidence. So funding science eventually will be fruitful, but it's a really slow and painful and meticulous process. And I wish to emphasize that in my talk. And with that, I'd like to conclude my talk, and thank you for your attention. And if you have questions, I will try to do my best to answer those to the best of my knowledge.
Yeah, that's great. Maybe we can do a few questions. I also want to alert you, so the next round of breakouts are starting as well. So there is a breakout session with variant syndrome. So if you have PSC or PBC in conjunction with AIH, Dr. Patel will be talking about that down the hall as well.
Yes, sir.
Great presentation. Quick question, though. You talked about THC being pro-inflammatory and CBD being anti-inflammatory. Can you describe that in a little bit more detail?
Wish I could. Wish I could. THC being pro-inflammatory, that is, we have preclinical evidence showing that. So we know macrophages have CB1 receptors. And THC is an agonist of CB1 receptors. So if you have THC in your system, that will bind and activate CB1 receptors. We know that resident macrophages in the liver, also in the pancreas, if they're activated via CB1 receptors, they will produce pro-inflammatory cytokines. Mostly that is IL-1 beta, TNF-alpha, and also some chemokines that will attract more immune cells to that location. That is well established in some models of liver cirrhosis, in alcoholic fatty liver disease, models of alcoholic fatty liver disease. But we also published a paper in Diabetes a couple of years ago showing that activation of CB1 receptors in the pancreas can cause the deterioration of insulin-producing beta cells. Now, that is specific CB1 receptor activation. If you consume marijuana, you will have all those other compounds, and particularly CBD, that will modify the effect of THC. So you can't directly extrapolate this, pharmacological, really specific data into cannabinoid product consumption. That's why we have so few solid evidence about cannabis and cannabinoids, because you have a lot of compounds in those products. So if you specifically activate CB1 receptors, you see these effects. If someone consumes cannabis, you'll have these effects plus all the other effects that will, all together eventually decide what happens in the body. But CB1 receptor activation specifically may have pro-inflammatory actions.
I'm sorry, I just have a quick question. So is THC, does it, maybe you as a physician you know this too, but does it harm your liver if you ingest it? When I was first diagnosed and my LFTs were very high, my doctor told me you could not ingest anything. You could only smoke it at that point. I live in a state where it's legal. But as my LFTs are normalized, I'm on regular medication, it's okay to ingest it. But how does it process by the liver, et cetera?
So those are more than one question, as I try to remember all of those. So if in the healthy population, if you consume a lot of THC, we don't think it's directly hepatotoxic. So I don't know about data showing that it's directly hepatotoxic. Having said that, we also know that in the healthy liver, you barely have any CB1 receptors. So it has a very low expression. So the THC is in your liver, but the receptor is not there. We also know that in certain liver conditions, that is not true anymore. You will have a lot of CB1 receptors in your liver and the immune cells that go into your liver. So what applies to a healthy population may not apply to a population that has ongoing liver conditions. So you can't extrapolate from the healthy population to the affected population. I can speak about preclinical data. If you induced, for instance, cirrhosis in animals via cholestasis, that kind of models PBC and PSC, and you add THC on top, you can aggravate the pathological conditions. But then again, those animals will have more CB1 receptors. So whether THC is toxic to your liver if you're healthy, and if you don't, consume doses that are close to, the EC50, to the lethal dose is 50, then probably not. If you have an ongoing liver conditions, that probably doesn't apply.
So from my clinical perspective, THC, again, no good models saying, at least in the clinical setting, of direct hepatotoxicity. There is a big caveat. So CBD, and he had talked about Apodolux as a therapy for pediatric seizure disorders. There is a dose-dependent response, and there is a signal that there is some liver toxicity at high doses. So the CBD usage from our paper, when we surveyed all of you patients in the past 3 or 4 years, the doses range from anywhere from 1 to 5 to 10 milligrams. The doses we're seeing in those reports of liver toxicity, which are rare, is still typically over 800 milligrams per day. Yeah, so
But
I want to
that's
give
CBD,
perspective.
right?
I'm sorry?
That's CBD.
That's CBD,
That's CBD.
correct. So I think no matter what, maybe we can paint a picture of honesty with your physician and how you treat and what you're taking. And one of the reasons we worry about CBD is it's metabolized through a certain pathway called CYP3A4. This is an enzyme that contributes, I think it's a 50% of the metabolism. The drugs that you all take for AIH are actually metabolized through that pathway as well. So theoretically, you can change some of the levels, and I think we talked about this in your talk, that may have implications for your disease and inflammatory control as well. So again, honesty, openness. I joke with everyone, all of our conversations on our Zoom support calls always ends up with discussion about CBD or THC. So we know it's out there, we know there's legal states, people are using it, but also that's probably more of a pointed exclamation point on how bad symptoms are that people are finding other alternative strategies to treat this outside their doctor's office. So maybe with that we'll end so we can move on. Thank you. Again, thanks for a great talk.